food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Can you talk about a little bit about your background and what you do?
I'm a professor at the Jackson Laboratory. I've been here for more than 25 years now. I love living in this part of the world. I was born in Maine, and I live right next to a national park, and there's a lot of benefits to it. I got my degree in mathematics. I went on to get a PhD in statistics, and I've always been interested in genetics. And I came to the Jackson Lab by circumstances of life and really became immersed in mouse genetics. Mm-hmm. So if I had to pigeonhole myself, I would say that I'm a mouse geneticist. And for the past 15 or so years, I've been focusing on metabolism and aging as my areas of interest, but I still retain that mathematics, statistics, data-oriented mindset. So a lot of my work is related to experimental design and interpretation of data.
Yeah. And many people might not know this, but it's interesting that you have the statistics background and then you went into the realm of biology and genetics. Genetics as a field, it's obviously a field of biology, but it has a very rich history of a lot of hardcore statistics. And I think some people might not realize like how hardcore the stats are that a lot of people, the chops and stats people have when they do genetics.
Yeah, genetics is a really quantitative field. I remember as an undergraduate acing the genetics course, and all the biology majors were dreading it. So it's wonderful. It suits me very well. It lends itself to analytical thinking, but it also throws some real biology at you there. I mean, biology is complicated and messy. And I will say before we get into it that one of the challenges of dealing with this, unraveling this story, is that it's complex. Mm-hmm. And of course, you want to tell a simple story so that, you communicate something, that it's effective. But I think the complexity is real. Yeah. And I kind of relish it. So,
yeah. And we'll get into that. It's going to be reflected a lot, I think, in this paper that we're going to discuss, just because and this is often some of the most interesting stuff where, I think you have results here that are counter to what most people would have predicted and defy obvious explanation. And we'll talk about some of that. For background, many people might not know what the Jackson Laboratory is, but it's a very sort of important node in the research world. So what is Jackson Labs?
Of course, the Jackson Laboratory is a nonprofit research institute. We're located on the Mount Desert Island off the coast of Maine, founded in 1929 by, moguls who came here for the summer and thought it'd be cool to start a scientific institute. They did. We're most well known for mice. We do, most of the researchers here work with mice as a model organism. Our focus is on using mouse models and genetic tools to investigate human health, really. That's in a nutshell.
Mm-hmm. And so, you've got this new paper out that we're going to discuss that's really interesting. It was quite a massive undertaking, it looks like. And it has to do with the relationship between dietary restriction and health and lifespan. So this is a mouse study. We're going to unpack it. You used a lot of animals here, and you used specific types of mice, and we're going to get into all of that. I want to give people a general base here, some background on some terminology, some concepts, and some limitations and methodological issues that are often inherent to this kind of work that, at least to some extent, you guys are getting around with this study. One of the things I want to talk about here is just mouse genetics at a basic level. So most experiments that are done in laboratory animals like mice are often done in inbred mouse strains. Can you talk about why that is and the types of mice you used in this study and why?
Okay, inbred mouse strains like the C57 Black 6J mouse that was developed here at the Jackson Lab were developed to ensure that experimental results would be more reproducible. Mm-hmm. An inbred mouse is created by a long series of generations of brother-sister mating, so that each of these mice inherits half of their genome from their mother and half of their genome from their father. And after many generations, those two genomes become more and more identical, so that in a sense, they're almost identical twins of themselves. Mm-hmm. And they can reproduce exactly the same genotype over and over again. So when we study an inbred mouse, it's really akin to studying one person. And they're also inbred, which is a little unusual in nature. There are some challenges that come with being inbred, and I know where we're going with this. So,
yeah. So on the one hand, having this relatively genetically homogeneous inbred mouse strain or laboratory strain of animals, that can be a feature with respect to research because it can give you more reproducibility. There's less genetic variability that's going to introduce variation and noise potentially into the results. On the other hand, as you say, it might be problematic from another angle to think about this because, inbreeding is not something you see in nature, and it's not necessarily going to, we might find some things that reflect not what we would see in nature in a more genetically diverse population that might inhibit our ability to generalize results from these particularly bred laboratory strains to other species, including even ourselves.
I think the key word there is generalizability. Mm-hmm. If we study C57 Black 6 mouse, at the end of the day, we're really limited in the scope of what we can say to just that particular mouse strain. And anything that we say about other mouse strains or other species like humans is extrapolation, and it may be true, but it's guesswork. So by working with the outbred mice, which I'll explain if you would, we increase the generalizability of our studies to at least more mice. Mm-hmm, mm-hmm. And we feel that that's going to increase our potential to generalize to humans, which is the objective here.
Mm-hmm. And in very simple terms, so what is outbreeding here? Is basically the opposite of inbreeding. So you're creating genetic diversity. Is there anything more we should know about that?
The outbred mice, the particular ones that I worked with in this study, were originally 8 different inbred strains that we mated together with one another, and we maintained them again for many generations. But instead of mating brother-sister together, we randomized the mating so that, just like people, the mother and father of the next generation are not related to each other. And that makes the genomes all mixed up, and it makes every mouse individual and unique. And that has some drawbacks because we can't put the same genotype in 2 conditions. It would be fantastic to do this study and have the same genotype on all 5 diets. But we'll do that. We'll figure out how to do that. But the outbred mice, really, I think, I don't want to say they're a model for human populations because they're not. In some ways, they're a better tool for genetics, but they have the kind of heterogeneity and diversity that you see in humans. And I think that's exceptionally important for aging because I think everybody ages in their own way. So each of these mice is going to age in its own way. Oh, and by the way, outbred means the two copies of their genome are not identical, and that gives them a feature called hybrid vigor. So they're really healthy, generally speaking.
So yeah, coarse-grained level, generally speaking, these mice sort of look and act obviously healthier. Presumably they live longer than the traditionally used inbred mouse strains as well, I would imagine.
Well, surprisingly, no. Their average lifespan is about the same. It's about 20 months, so a little shy of 2 years average. But what's different is the variance in the lifespan. There are outbred mice an inbred mouse will have a lifespan that's pretty within a window. There's a lot of randomness to it. And they almost always die of the same causes. Like a C57 will almost always die of a lymphoma. They have their own built-in genetic time bomb, so to speak. And the diversity mice, well, they display all kinds of lifespans and different causes of death. And we've had outbred mice live as long as 4 years and 9 months, which I'm going to say it in dog years is 140. It's a bit of a joke.
So I think what you're saying is the average lifespan of these outbred mice is about the same as the inbred mice, but there's more variability in terms of how long they live population wide, more variability in terms of the ways in which they succumb.
Yeah, I could have said that. Thanks.
So basic concepts here. We're talking about dietary restriction and its impact on health and longevity in these outbred mice. We've got calorie restriction, we've got dietary restriction, and then we've got intermittent fasting. What are the essential differences between these three things?
Well, yeah, the language, we try to be consistent within our paper, but it's not always consistent across all the literature. So what I mean here by caloric restriction is we fed the mice a standard chow diet that's a healthy diet. We figured out how much a normal adult mouse would eat, and we reduced the amount of food that we provide them by either 20% or 40%. So they're getting 80% of the food they would normally eat or 60% of the food they would normally eat. So those are the caloric restriction. And intermittent fasting, there's a lot of ways to do intermittent fasting. Probably most of your listeners are going to be familiar with the 16-8 paradigm. It's very popular these days. People restrict their eating to 8 hours a day. We did something a little different here. We did intermittent fasting where we removed the food from the mice on Wednesday and we gave it back to them on Thursday or Friday. So they either went for 24 hours or for 48 hours without food. And I know people listening are thinking, well, I could do that. I could go 24 hours without eating, but you're not a mouse.
Yeah.
And one of the things I want to emphasize, and one of my concerns about publishing this paper is that people will look at these diets and they'll think, I should do that. And I would highly advise no. You will not tolerate 40% caloric restriction. People barely tolerate 20% caloric restriction. When we fast the mice for 1 day or 2 days, it's a little hard to say what the equivalence is, but it would be like you fasting for 2 weeks.
Yeah, because they're small animals, their metabolic rate is different.
You wouldn't want to do that.
Yeah,
exactly.
Yeah.
So my point is these interventions, CR and IF, as we abbreviate them, they're pretty extreme. And we needed a term to refer to them both at the same time so that we didn't have to write CR and IF every time.
So these are all forms of dietary restriction.
So they're both dietary restriction. And I use the word dietary restriction very broadly to refer to almost any kind of restrictive intake, whether it's timing or calories.
And obviously everyone is inherently interested in health and in longevity. But I think the point you're trying to make here is we have to be very cautious about jumping to conclusions about humans here because, 1 day of fasting in a mouse is not equivalent to 1 day of fasting in a human. We're talking about fairly strong interventions here. And I think that would imply that all of the results we're going to talk about are right. We need to keep in mind that these are strong interventions. And so any more mild forms of fast. So if I fast for 24 hours here, I should expect, if anything, weaker effects than what you saw in the mice, for example.
I would not argue with that. And I would also say that there can be benefits to reducing calories and to fasting.
Mm-hmm.
We just need to be a little careful. And what we're trying to do with the mice here is to push the envelope because we're interested in the fundamental biology. Ultimately, we want to understand the mechanisms. And one possible outcome of these studies down the road would be to understand the mechanisms and find different ways to intervene so that we could use our understanding derived from calorie restriction to find other routes to improve health.
Mm-hmm. And so I want to give people a basic sense before jumping into the results themselves of where this field, broadly speaking, sort of stood and what some of the motivation to do this work was. So obviously, people have been talking about calorie restriction and dietary restrictions of different kinds for some time. Intermittent fasting is very popular. And I think sort of a headline summary of this field is that in general, people often find that dietary restrictions like this tend to result in health benefits and increased lifespan. Is that basically what the thinking has been, and how does this tie into what is known and unknown about what was actually causing these lifespan benefits from restrictions?
Okay, it's a little nuanced, but let's start with the dietary restrictions. Caloric restriction in particular has been known to extend lifespan in rodents for 100 years.
Yeah.
We also have seen research in the past decade or two suggesting that not every genotype. So remember, if we look at inbred mice, if we calorie restrict C57 Black 6, they live longer. But it wasn't really clear that all mice would benefit from caloric restriction. And I think that was the biggest motivator. Well, I did this. It was the biggest motivator for doing this study without bred mice to ask the question whether caloric restriction would be universally beneficial or whether there might be subsets of animals that really didn't benefit from it in terms of lifespan.
Yeah.
Lifespan is really is a measure of aging, and I would like to, if you would help us come back to that point in a bit. The question I would ask, though, is do you want to live as long as you possibly can, or do you want to be healthy while you're alive?
Yeah. Health span as opposed to lifespan.
So we did focus on lifespan as our primary singular outcome. But as you know from looking at the study, we measured everything under the sun related to health, everything you could think of. And in particular, these interventions improve metabolic health. And this is a really good thing for people because we have such a high prevalence of metabolic disease. And we saw these improvements in metabolic health in the mice on the diets. So extend lifespan, improve health with some caveats.
So another thing that you did here is not only are you using outbred mice, they're more genetically diverse. You used a lot of mice. This study involved almost 1,000 mice. Obviously, this gives you statistical power. You also needed to do that more just sort of by necessity because you don't have genetic homogeneity. So there's going to be more natural variation from mouse to mouse. You're looking at 5 different cohorts of mice that get to eat whatever they want all the time. You've got mice that have, 1 day of fasting imposed on them, mice who have 2 days of fasting imposed on them. And then you've got the 20% and 40% restricted groups. They can eat as much as they want whenever they want, but there's this limit on the total available calories to them. And as you said, just keep in mind, everyone, we're not going to list every single thing that you guys measured, but you measured a lot. Can you give us just a basic sense for the diversity of measurements and biomarkers and things?
I will talk about the scale that the mice from the first cage that we filled until the last mice died spanned almost 8 years.
Wow.
At the peak of the study, we had 7 full-time technicians just managing the phenotyping schedule. Um, we really learned a lot. We did all kinds of clinical assays. We weighed them every week. We put them in an NMR, an imaging machine to look at their body composition. We put them in metabolic cages for a week at a time to see their circadian rhythms and their metabolism, their activity. They went on wheels. We did multiple blood draws. They got echocardiogram. I'll stop. You get the idea.
Yeah. Everything, like everything under the sun.
Yeah, why not? You're just going to do this once.
Yeah. So very thorough, very, very massive study, 8 years of time, it sounds like. One last piece to mention maybe is I think you looked at female mice only. Is that just to decrease? Is that true? And was that just to sort of decrease?
We looked only at female mice.
Mhm.
And I think that's a weakness of the study. But I'm going to add that there have been so many studies that have been done only on male mice that I feel like it was fair to tip the teeter-totter the other way.
Mhm.
One of the issues with the diversity mice is that they are genuinely real wild mice.
Yeah.
And in the wild, female mice tend to live together rather well, and male mice not so well always.
Mhm.
It doesn't happen all the time, but if you leave male mice together for a long time in a cage and you deprive them of food, they fight.
Yeah.
And once the mice start fighting, they get injured and you're obliged to euthanize them.
Yeah.
And, we felt that.
But anyway, so you've got almost 1,000 female mice in this study. What's the first basic result here? So you've got these 5 different groups of mice that are on 5 different feeding schedules, basically. What's the headline result here in terms of how raw lifespan differed between the groups?
Well, the basic result is that these dietary interventions all extended lifespan. And you might say to yourself, that's not a surprise, but I feel like it was somewhat surprising to me, especially with the 2-day intermittent and the 40% caloric restriction, because I felt that one of the likely outcomes is that these interventions would have such severe detriments that we would shorten the lifespan of some animals. And honestly, I didn't see that. There was no indication that there was any fatalities due to the interventions. So that was a good outcome for us. But in the strictest sense, as a statistician, I think what we've demonstrated here is that when we assign these diets at random, they extend the lifespan. And because of that, we have a causal relationship. Dietary intervention causes lifespan extension. And I'm looking at my notes here because I want to get this right. But I want you to think of a little circle with diet written in it, and a little circle over here with lifespan written in it, and an arrow that points from diet to lifespan with a plus sign on it. And we established that. But why do we measure all that other stuff? I'm sorry, I'm not supposed to ask you questions.
Well, I mean, so I mean, there's some natural questions here. Is yes, these mice may be living longer, but are they more sickly? Are they more frail? Do they have less energy? Are they less healthy in some sense based on other measures we might care about?
What we saw, and when you let's talk about the 40% CR mice because they are so extreme. All the mice on interventions lost weight. The 40% CR mice lost weight continually through their lifetime. They lost fat mass, they lost lean mass. This is not good. You would not like that. But when we look at the mice, we handle them. They're really quite healthy. They don't have any obvious deficits. They're not sick. In fact, we know that we reduce the incidence of palpable tumors, so they're likely not getting cancers while they're on these interventions. Their blood glucose stabilizes. They don't get obese. I could go on and on. But when we looked a little deeper, especially when we looked at properties of the immune system and we looked at the red blood cells, there were definitely indicators that these interventions are not universally good. And I think, whenever you manipulate a complex system in any way, you're going to have good and bad, right?
Yeah. And maybe something important to keep in mind as we go through some of the details is, the mice in these groups did have lifespan effects. So 40% calorie restriction resulted in quite a substantial increase in lifespan. But as you said, and as we'll keep in mind as we go through some of these biomarkers and other things, I want to emphasize too that these mice are living in assisted living for their entire lives. They don't have to worry about dense packing in large groups that might result in pathogen transmission. They don't have to worry about getting eaten by an owl or a coyote or a predator. And so I would imagine that some of the things that we talk about, even though these mice live longer with these dietary restrictions, such mice under such conditions, if sort of transposed into the wild, might actually live shorter lives for various reasons we can imagine.
This is the generalizability question.
Mm-hmm.
I can say that under these conditions, the dietary restriction extends lifespan. You were right. There are no predators. There are no pathogens. They live in a clean facility. We do house them in large groups because they're social. They like to be, not packed in, but they like to live close to one another. Yeah, they're healthy mice, and they're healthy mice because they get a good diet and we take care of them. Veterinarians look at them every month, and yeah.
Mm-hmm. So getting into some of the results here, can you give us just a little bit more detail on how some basic measures changed across the lifespan in the different groups, body weight and body composition, adiposity, things like that?
Yeah, we saw some interesting differences between caloric restriction and intermittent fasting. And the one that really struck me is that mice who are feeding ad lib, so all you can eat, which mice self-regulate, they do a good job. They have healthy diets.
Mm-hmm.
They tend to grow and they gain lean mass almost throughout their entire lifespan.
Mm-hmm.
And the intermittent fasting mice, despite their weekly deprivation, if you would, gain lean mass. They lose some fat mass, and so they're leaner, but they don't lose that muscle. They don't get smaller. Caloric restriction mice lose everything. They lose lean mass. They get smaller. That's one thing we noticed. Another thing we noticed, and that is, I need to talk about red blood cells for a minute.
Mm-hmm.
You want to give me permission to do that?
Yeah, absolutely. Yeah.
Red blood cells are the most common red cell in your body. You've got trillions of these things, okay? Mm-hmm. And they not only do they deliver oxygen to your whole system, but they do all kinds of biochemical things while they're flowing around through your circulation. And a red blood cell lives about 120 days in circulation. And the way that happens is as red blood cells age, they tend to get bigger. And your spleen is constantly monitoring the red cells, and it'll identify the big ones and filter them out. So it's keeping the red cell population healthy. Mm-hmm. So one of the measures that we take on these mice is we look at their red cells. We know how big they are. We know how much hemoglobin they have in them. And we know their distribution. We know how big they are. And what happens in especially the 2-day intermittent fasting, but also in the 40% CR mice, is that distribution gets wider. And that tells us that the spleen is not doing its job efficiently. It's not bringing out the red blood cells the way it should be. And this measure, RDW, it'll be on your report from your annual health, because it's typical clinical measurement from a complete blood count test, is already known to predict lifespan in humans. And here we're looking at interventions that extend lifespan, but they also increase RDW, which reduces lifespan. And and this is a pattern and a theme that comes up over and over again. And remember, diet, arrow pointing to lifespan with a plus sign. Now, that arrow has hidden in it all kinds of things, and that's why we measured all kinds of things. We want to pull that arrow apart and find out, well, what are the steps along the way from diet to lifespan? And one of those things is RDW. So you picture an arrow from diet to RDW with a plus sign, but then RDW to lifespan with a minus sign.
Hmm.
It's contradictory.
Right.
And we saw this again and again. The diet makes something worse, but the diet extends lifespan.
Hmm.
Um, we
Yeah.
saw that. Yeah,
And
go on.
I think we'll come back to that, but is is the punchline here that that is just mysterious at this point?
Is it mysterious? No, I don't. I
Hmm.
don't think it's mysterious at all. I think that the diets don't have a singular effect.
Hmm.
They do lots of things to the system as a whole. And if you think about what lifespan means, lifespan is how long the animal lives. Animals are going to live for a period of time that is to a large extent random, but it's also determined by many, many things. And they die of the first thing that kills them.
Hmm.
And there are many ways to die.
Yeah.
And,
Yeah.
if you have failure related to your red cells, or if you have other kidney decline or cancer, so diet affects lifespan, but lifespan is a complicated thing.
Hmm.
We want to know what are the components of lifespan that are affected by diet. And some of those are affected in a good way, and some of those are
Hmm.
affected in a not so good way.
I see. So for simplicity, if we think of our arrow, it's going from diet to lifespan. Let's just say
So
inside of that arrow, there's actually 100 different factors, 100 different ways,
at
things that could
least you wrote
break
it.
at least.
Yeah.
But for simplicity, let's say 100, and some the dietary restrictions are having some pattern of effect on all or many of those things. And so even if some subset of those things goes down or becomes more negative, it's a bad effect. There might be a boosting or an increase or a positive effect on some greater number of other things, such that the net effect is that as a population, you tend to see a lifespan extension.
That is the key as a population. So let's clarify something we said earlier. When we say the dietary restrictions extend lifespan, does that mean that you as an individual could go on dietary restriction and you're guaranteed to have a longer lifespan? No. It would, on average, increase your chances. But when I say on average, I'm really talking about a population, and in this case, a large group of mice on dietary restriction means, for whatever reason, some of them manage to live longer. Um, so it's not that there's a guarantee that this intervention will extend lifespan, but it's going to do lots of things. And depending on luck of the draw and genetics,
Mm-hmm.
maybe it's going to extend lifespan or not affect lifespan at all.
Mm-hmm. Mm-hmm. I want to talk a little bit about, for a few minutes here about metabolic effects. And obviously, when you restrict an animal's diet, whether it's the calorie restriction by 20 or 40%, whether it's the 1 or 2 days of imposed fasting that some of the other mice got, obviously this is going to have metabolic effects. It's going to influence things like glucose homeostasis, like insulin resistance, like daily energy expenditure. Can you talk about some of those variables, some of those big metabolic variables, things that we knew would change, and how they actually changed across the groups and what the relationship was with lifespan?
Okay. When we talk about metabolic health, um, you have to keep in mind that the situation for modern humans is very different from the situation with mice in a mouse room.
Mm-hmm.
Um, it actually, there are a lot of scientists who would contend that caloric restriction works because we cure obesity and all the associated detriments that go along with obesity.
Mm-hmm.
Mice, at least the diversity mice, they don't they don't necessarily get obese. Some of them do, some of
Mm-hmm.
them don't. They're all
Mm-hmm.
different.
So you're just
Um,
saying the mice that had free access to food for their whole lives, for the whole duration of this experiment, they gain weight on average
they
throughout their
gain weight.
lifespan, which is normal for animals as they get older and grow and everything. But they weren't becoming obese.
Right. So out of 1,000 animals, 160 plus of them on the ad lib diet, we had one overt diabetic mouse with a
Hmm.
blood glucose that went up over 300.
Hmm.
So they're generally healthy, but let's come back to what the metabolic effects are.
Hmm.
So the diet causes a reduction in body weight, which is largely fat, so they're leaner. Um, their glucose levels go down. Um, what else? Their body temperature goes down. I would say that's not a desirable thing. They're cold.
Hmm.
Uh, their activity is interesting because when the mice are on IF and they're not being fed, their activity is low, their energy expenditure is low, but then they refeed and they have high activity. If you go in the mouse room and look at the mice, the restricted mice are the active ones. They're popping up and down and jumping around. And that's because they know you're there and
Mm.
they know where food comes from. So
They're getting excited by the presence of the human that predicts food.
yeah, so even though the impression is that they're more active, they're actually less active.
I see.
Although
So,
I will say that 40% CR mice retain their activity levels throughout life really consistently.
hmm.
Did I digress too much? But
No,
the
I mean,
obesity goes down and the blood sugar stabilizes, and these all sound like really good things, and
yeah,
they are in a human context, they are good things.
yeah. So you restrict the animal's diet. 20 to 40% of calories are restricted, or they're on this 1 or 2 day a week fasting schedule where there's no food access. You're saying you see a lot of expected changes, stabilization of glucose levels, decreases in energy expenditure, decreases in body temperature, things that you would expect to change that go in the expected direction. And yet,
Okay, so this comes down to the statistical details of how we understand the data.
mhm,
But let's suppose that the diets are working by reducing obesity.
yeah,
Then what should happen is if I look at animals within one diet,
yep,
this is stratification.
mhm,
If I just look within one diet, if the reason that diet is extending lifespan is due to reduction in obesity, then the animals that lose the most weight should live the longest.
yes.
That's the expected result. But what we actually see is the animals that lose the most weight live the shortest.
Mhm. So let's say that again. So within a group, so you're restricting the calories that these animals can take in.
Yeah.
So you impose that thing on a group of mice, and they live longer as a population. The thinking is, okay, well, you're reducing calories. Naturally, the mice will be leaner, they're less fat. And so, if that is the reason they are living longer, then within that group, the ones that lose the most fat should tend to live the longest, and the ones that lose the least fat should tend to live the shortest. But what you're saying is that you observe the opposite.
The opposite. Yeah. And
So
this
within a
is,
group, the ones the ones that are the least lean are tending to live longer.
yeah, I would say that these diets, they cause extended lifespan. They cause reduced body weight. But if we look within a group and we ask who lost the most weight, they have shorter lifespans, and that is telling me that the reason for lifespan extension is not lost weight.
Hmm. What about body temperature?
It's the same story for body temperature.
Hmm. And I guess
Um,
naturally, sort of maybe a naive prediction here would be like, okay, if I'm restricting calories, the body has just got less metabolic activity going on. I'm slowing
yeah,
things down. I'm cooling
I'll
things
last
down,
longer.
and therefore the engine will burn hot less often, and I'll live longer.
Mm,
Is that
That
basically how people would think about it?
that is how people have been thinking about it. Yeah.
And then you see the opposite within a group. It's actually the ones that have the higher body temperature that tend to live longer.
hmm Yes.
Hmm.
And I'm going to put my interpretation on this.
Yeah,
It is that I think the diets are stressful.
they're stressful.
Yeah. And obviously, just think about it. Someone takes away half your food. This is
Yeah,
stressful.
yeah,
The diets are stressful. Stress has some benefits. Stress has some downsides,
yeah.
like our,
You want some stress, but not too much.
yeah. So the animals that better tolerate the stress, they lose less weight.
Mm. which
You
makes
know,
sense. Yeah.
yeah. And the animals that are more sensitive to the stress, they're going to lose weight. I think weight loss is a really good indicator of stress in a mouse.
Hmm.
When they're stressed, they're going to lose weight. They burn more energy. And so I think that animals coming into a diet group that are already intrinsically more resilient deal with the stress of the diet better and they
Hmm.
gain the most benefit.
Hmm.
And the animals that come into the diet and they're already maybe not that robust are going to be hit harder by the diet. They're going to lose weight and they're naturally going to have a shorter lifespan.
Hmm.
So,
Did you
uh,
have any way to measure something like stress resilience by other means?
measuring stress resilience is a big open question for the field. You can imagine that there are some ethical considerations. We're very serious about how we handle animals, and
Hmm.
of course, we want to avoid stressing animals. The way we measured stress resilience was actually after the fact. During their lifespans, I told you how much phenotyping, how much measurement we made on these animals.
Hmm.
There was a period once a year where they spent a whole month in the facility where we did all these procedures, where we weighed them and put them in
Hmm.
the NMR machine and put
Yeah.
them in metabolic
You have to
cages.
pick them up. You have to disturb them. It's
Yeah.
stressful.
And part of that, I think the biggest stressor for them is to be separated from their cage mates,
Hmm.
because
Hmm.
when they're isolated, they're looking for their cage mates. They know something's disrupted. They're not in their home cage. And what we noticed is during that month, almost all mice lost weight.
Mm.
No surprise, it's stressful.
Yep.
So I thought, well, how much weight did they lose? And so we went through and we weigh them all the time. We weigh them at least once a week. So we went through and we blocked off that month from beginning to end and measured how much weight did they lose.
Hmm.
What we found is that the more weight they lost, the shorter their lifespan would be. And this was true within every diet group. And
Ah.
again, like I was saying before, I think this is a measure of how resilient the animals are to stress.
Yeah. So the idea
Um,
here is what's nice about this is you're finding something that cuts across all of the conditions. So within every condition, the mice might have access to food at all times. They might have 40% of their calories taken away. They might be on this intermittent fasting schedule for 1 or 2 days. Within each group, when you do this month of testing, you could measure how much weight they lost, and they would all lose weight because that's stressful and it just results in weight loss. But you're saying the ones that lost the least weight, which implies that they're probably more stress resilient, tended to live longer no matter what the feeding condition was.
yeah, isn't that amazing?
Yeah.
I, I
And
love it.
it sort of really does kind of start to twist and change a little bit how you think about things like dietary restriction. As it's not merely changing the pattern of food that an animal eats. It is a form of stress that must be dealt with by the organism.
Mhm. Agreed. And stressors can have beneficial effects because you think of a mild stressor is enough to stimulate your body's homeostatic response. Your
Mm-hmm.
body will respond to a stress by bringing compensatory mechanisms on board. And if you stress an animal severely, you're going to do damage to them, and that's not good. So we avoid that. But if you stress them a little bit, they and I can't prove this with the experiment that we did. We're going to do some more. But I think the diets could potentially also trigger these homeostatic responses and induce more resilience than background,
Mm-hmm.
if that
Mm-hmm.
makes sense.
Yeah. And so one of the things that you found as sort of like an overarching thing is like figure 3, for example, it's titled Health and Metabolic Traits Change with Age and Diet, but are poor predictors of lifespan. So you guys measured all sorts of stuff: cardiac output, grip strength, prevalence of tumors, just wheel running activity, all sorts of stuff. And you found that many of these things were changed by diet and did change across the lifespan, which were unsurprising, and yet they poorly predicted lifespan. Can you give us some examples there, and maybe sort of what sort of the overarching result was in your view?
Yeah, cardiac output. I forget which way it goes. Let's see if I have actually have figure 3 somewhere.
Um,
Did we look at?
yeah, I've also got it right here.
I can't even read this thing. I pored over every word for so long. I can't even do it. What did we put on there? Oh, fasting glucose, body
Yeah.
temperature, and frailty. I lost the question. Sorry,
Okay,
guy.
hold on. Let me pull this up here. You measured basically you measured a bunch of health and metabolic traits,
We did.
and some of these change with age, as you would expect. Animals get more frail with age. You expect something like grip strength to go down with age, I imagine. But nonetheless, even though they changed as a result of the dietary interventions, even though they changed across the lifespan, they were often poor predictors of lifespan.
Right. Let's go back to the arrows. And if we think of it
Yeah.
as the diet is causing changes in these traits,
Yeah.
so the diet has an arrow that points to cardiac output. It has an arrow that points to grip strength, and it affects these things, but there's no arrow from grip strength to lifespan. There's no arrow
Yeah. Yeah.
from surprisingly cardiac output to lifespan. So the diet has lots of other effects that are not intermediates for lifespan.
Yeah. Yeah.
It's
Yeah, and
hard.
they appear not to be intermediates, even though many people would have expected them to be.
Yes.
Yeah.
And again, improving metabolic health is a good thing, especially for people.
Hmm.
Improving cardiac output, it's a good thing. So it's do you want to live as long as you possibly can, or do you want to be healthy while you're alive? And I
Hmm.
think some of these effects are definitely in the healthy direction. They're beneficial and they're good, and they don't necessarily extend lifespan. So we shouldn't equate good health with long lifespan.
Yeah. Yeah.
That's a punchline.
Hmm. Were there any things that looked like? Were there any things that were clearly associated with longer lifespan that you would interpret as being like less healthy or less desirable to have if one is alive?
Oh, absolutely, the weight loss.
Yeah.
Uh, yeah, I think that's the big one.
Yeah. Just losing all of that mass, including that lean mass.
Yeah.
Yeah, and I mean, so again, we don't want to anthropomorphize, but give us a set. So remind us again how much. So like in the 20 or 40% calorie restriction group, that's quite a severe restriction. Remind us how much fat and how much lean mass was lost there. Quite a bit.
Oh, that is a good. They were about half the size that
Yeah,
they would be on an ad lib diet.
yeah, yeah. So
Oh,
in a human being, that would be, that's
that
imagine
would
being, I
be
don't know, roughly half as big as you would have been.
yes, and not desirable. Just put
Yeah,
it that
yeah.
way.
And do you think so? So things like that in these conditions, we mentioned this before, under these conditions, these things are associated with longer lifespan, but it's very easy to imagine that such a mouse living out in the real world outside of assisted living, those things would actually be a huge liability.
Well, let's talk about the immune system for a minute. And I'm
Mm,
on thin ice here, and I really did rely on my immunologist colleagues at Calico and at the Jackson Laboratory to help us understand what the immune system changes were.
yeah.
And when animals went on these diets, the caloric restriction, one thing we noticed is reduced inflammation. And this is
Hmm.
definitely a good thing.
Yep.
We saw fewer inflammatory macrophages, and we presume based on that we have less inflammation. But there's a whole population of cells that are called NK. They're natural killer cells, and their job is to go sniffing around for viruses, tumor, bacteria, and set off the alarm bells and start the process of attacking the virus or getting rid of the bacteria or addressing the tumor. And these cell populations were badly depleted by the diets. So a mouse in the wild would be exposed to all kinds of pathogens, viruses everywhere. And if that mouse were deprived of calories to this extent, its immune system would not function well and it would not survive.
And is the basic interpretation there simply that, is it basically a common sense interpretation? The immune system is important and it's metabolically expensive. There's a lot of fuel that needs to run an immune system to deal with an infection, clear it out. And therefore, if you substantially restrict the diet like this, it's probably not surprising that the immune system is not going to be able to run at full throttle.
I think that's absolutely true. It's accurate. And there's another thing to know about the immune system, and it's a very knife edge thing because when your immune system kicks in and it's fighting an infection, it does a lot of damage. So there's a real fine balancing act between how reactive your immune system is. We know with COVID, for example, when people would die of this, it would often be because of the inflammatory effects that are the sequelae of the immune reaction and, not a direct result of the virus. So your immune system has to be kind of in this just right place. And in the dietary restriction, at least that component of the immune system represented by the NK cells is kind of down. It's not up to speed, so it won't protect them from viruses. The flip side is they don't have inflammation.
Yeah.
Because their immune system's not overreacting to things. It's not.
Yeah. So they're probably on the one hand benefiting from lower levels of chronic inflammation. They're less likely to have an immune system that's inflaming the body too much in response to an injury or something like that. On the other hand, they are more susceptible to infection.
That's what I would guess.
Presumably, if you were to put them out in the real world.
Everything is context.
Yeah.
The mice live in SPF and we keep it pretty safe and clean for them.
So there's another sort of major result here that I want to talk about. So it was really cool. You were using so many mice in the study for such a long time. You had these genetically diverse mice. They aren't the inbred mouse strains that are typically used in experiments. So now you can look at actually genetic variability as a variable here. You can see how things vary as a function of the genetic distance between individuals within each of these groups. And there's a result here that I personally do not find very surprising at all, that I think is very important. I think some people maybe will find it surprising. Some people might find it, a little upsetting or something, sobering maybe. And obviously when we're talking about this stuff, dietary restriction, these are interventions. In human beings, we can choose to do things like intermittent fast and calorically restrict and go on diets and things like that. And the whole point is we want to use, we want to choose to do certain things that are healthy for us, that are beneficial for us. But you've got a result here that speaks to just the role that raw genetics played here. What was the relationship between the genetics of individuals and how long they lived?
Okay, this is a tricky one to navigate. So let me try and say, when we talk about nature versus nurture, genetics versus environment, it's always both.
Yeah.
And it to different degrees. And these mice are in really controlled environments. The temperature never changes. They get good food. The same thing happens. There are no pathogens, no predators. You pointed these things out. So those constant environmental conditions really raise the impact of the genetic portion of the equation. I would guess, and I don't have to guess because the data are out there, that the heritability, the proportion of lifespan in humans that's attributable to genetics is smaller, much smaller than the 24% that we saw here in the mice. Why is the genetic effect of interest to us? We can attribute 24% of the variability in the lifespan to genetic factors.
So almost a quarter, which is quite a lot, right?
Yeah, it's a lot. It's really, it's a whopper. and only, I forget the number, but only 7 or 8% to diet.
Mm-hmm.
Remember, everything's context dependent. So in this context, but the genetics gave me a ruler to measure how big the diet effect is. So remember, if you go on one of these diets, it's not a guarantee that you're going to live longer. It
Mm-hmm.
improves your odds, so to speak. Um, and how much is it contributing relative to having a 90-year-old grandmother?
Mm-hmm.
And you really want that 90-year-old grandmother.
Yeah,
There's,
that's what this
there's
result is
nothing,
basically
there's
saying.
nothing you can do about that. You can't choose that.
Mm-hmm.
Um, this is not, not your fate, though. There are a lot of things you can do within the hand that you're dealt to make yourself healthier and live longer. But it is what it is, and it's an important component
Mm-hmm.
of. And there's a third component here, and that is everything else that we don't understand.
Yeah.
I'm going to say that's 70% of it.
Mm-hmm. Mm-hmm.
I'll call it noise for today, but it's the randomness of lifespan.
Yeah, just like because there's so much in biology that is dependent on stochasticity. Just
The
something happens to go one way versus the other.
the bumps in the road.
Yeah, 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 and that's important to keep in mind. So I don't know if you could unpack this from this study, but I want to talk a little bit about the possibility of this. Um, is it possible that in different subsets of mice, for example, within each group, perhaps some mice with certain genotypes really benefit from caloric restriction with respect to lifespan, and maybe it's also possible that other subsets with different genotypic makeups are actually harmed by it. Is that possible or plausible here?
This is absolutely plausible, and it's one of the things that we intentionally look for.
Mm-hmm.
Can we identify genetic loci that would separate mice that have longer lifespans on diet from shorter lifespans on diet? And we looked, and I would say, we might need larger sample sizes, which may not be feasible. But I also think that those genetic determinants they're complex and they're kind of distributed all around. They're spread everywhere. We found one genetic locus that achieved the threshold for statistical significance, which is a pretty high bar. It's on mouse chromosome 18, and what I find fascinating about this genetic locus that is predictive of lifespan and has a pretty substantial effect, almost as big as the diets, is it's also a locus for RDW. So now we have a location in the genome. We know that this location has some variability. It's different mice have different flavors, and the mice with one flavor that I'll call castaneus, because that's the name of the mouse strain that, the bad allele is the cast allele in this case. And these mice
Remind people what RDW is.
RDW is the red cell distribution width trait that we talked about earlier, that's related to shorter lifespan. So in addition to diet affecting RDW and lifespan, we have a genetic locus that affects RDW and lifespan, which is why I keep coming back to that trait. And I don't think that RDW is a measurement that we make, but it does tell us that there's something about the maintenance of red cells that's really important, both in terms of genetics and in terms of response to interventions and how these things affect lifespan. I might have got off the rails there, but there's a whole 75% of the genetics is still unexplained. We have this one locus that has a very large effect. We see it. Yay. But most of the genetic variability is distributed all around the genome in lots of tiny little effects.
Yeah.
to me, this isn't surprising because, if there were large effects on lifespan segregating around the genome, evolution would have cleaned that up. It wouldn't, they wouldn't persist.
Mm-hmm.
So you expect a trait like lifespan or body weight to have lots of little tiny effects everywhere, which
Mm-hmm.
makes genetics interesting for a statistician, maybe disappointing for someone who hopes to find the gene,
Right,
the
right, right.
lifespan gene. It's in
Yeah.
there.
Yeah, it's not there. There is no one thing.
No, no.
Not even close. Um, what personally surprised you most about any of the results that you got, or defied your expectations from what they were when you went in?
I should have been prepared for that question. It's a good one. I really think it has to be the thing that we already discussed, that the paradoxical effect of weight loss.
Mm-hmm.
It was really kind of obvious up front that
Mm-hmm.
the diet causes weight loss, and that
Yeah.
should improve health, and that should improve lifespan. And I think that disconnect was surprising to me, and I'm sure it helped us get this paper published because it seemed surprising to everybody.
Yeah. I mean, and it's perfectly logical. I mean, we have an obesity epidemic. Basically, there's lots of opinions here, but at a high level, right, people are just eating more than ever in most cases. The obesity is bad for health, bad for lifespan. And therefore, if you counteract obesity and metabolic dysfunction more generally by restricting calories in an environment where most individuals are in calorie surplus, then it's logical to think that the lifespan extension is coming from that. But that's not the case here. As you said, in each group, even though they lost weight and they had some of these metabolic effects that you would expect, that was not what was responsible for the lifespan. It was actually the heavier animals within each group that were living the longest for that group.
You got it. I did want to just go a little further on the genetics
Mm-hmm.
discussion, because I think people have this fatalistic sense about genetics.
Yeah.
And I want to say still, I'm concerned about people reading the paper and going on these ridiculous diets, because the diets we put these mice on are really extreme. But there are a lot of things you can do. So I want to say, if your grandmother gives you advice, don't smoke, you know all these things, right? Exercise is big. Eat right, sleep, go to the doctor, nurture your relationships, and enjoy life. All these things are proven to improve your health and your lifespan. And if your grandmother's 99 years old and she's telling you all this stuff, maybe she did take her own advice or not. So, but it's a bonus to have the 99-year-old grandmother. But within the hand that you're dealt, within the constraints of your genetics, there's a lot of leeway, and there are a lot of healthy things that you can do. And this has nothing to do with my study, but it has a lot to do with how I understand aging and health.
Mm-hmm.
And it's personal, but I think it's good general wisdom.
Mm-hmm.
So,
one thing I want to, yeah. I mean, one thing I want to there's a couple things I want to come back to. I want to talk about the stress resilience piece a little bit more. That seems like a very important piece here. I mean, do you agree with that? Do you think that's part of, like, would you say that's a central finding here because it cuts across all of the groups? And where would you speculate or hypothesize that we could discover more about what's actually going on there at a mechanistic level?
Yes, the stress resilience. I do think that we could unravel some of the genetics of that, and I'm interested in how an individual can improve their stress resilience. I think all the grandmotherly advice that I just gave is going to improve your stress resilience.
Mm-hmm.
No, I'm at a loss.
Do you think, one thing that, like, I would like to just riff on, as you mentioned, you use the female mice, they're housed together. So there's social conditions here. Do you think that's possibly or plausibly playing into this? The fact that these mice were co-housed, they weren't lonely, they had some kind of social network. Do you think, the where they were in terms of social rank, the number of mice they had good relationships with, so to speak, do you think that could likely factor into what their stress resilience levels were, or something like that?
Yeah, I think it's huge. And we got special dispensation to house mice in larger groups than
Mm-hmm.
typical.
Mm-hmm.
One thing I had in mind when by the way, we put the mice in a large format mouse cage, which is larger than the normal cage,
Yeah.
and we put 8 mice per, so that there were 8 per group, and typically we'll do 4. You never want to singly house a mouse; it's just too stressful. But we typically do 4, but I thought 8 would be good. And one of my thoughts on this was that as these mice age and they're going to die, there's going to be the first mice that die, and then the second and third, and then there's going to be a last mouse. And that last mouse is singly housed by definition. You really can't rehouse them after
Mm.
it's, they have their friends and their social network and they don't want to change. So I wanted to have not too many singly housed mice at the end of their lives. And we are interested in this question. We have some data, and I can say that those singly housed mice, the last mouse doesn't live as long as the mice that have the social structure around them. But it's a bit of a tricky question. So, because they're also the longest lived.
Yeah, yeah. And the immune system results are also interesting and worth coming back to a little bit. So, you had these immune system changes that were induced by the dietary conditions. In essence, these significant caloric restrictions were, impacting the immune system in significant ways that would probably leave the restricted mice more susceptible to things like disease if they were living out in the wild, where they would more likely encounter pathogens. Do you think it's likely or plausible that the levels of dietary restriction that health conscious people are often imposing, things like 16/8 intermittent fasting, things like 1 or 2 days of fasting per week, do you think those restrictions self-imposed in humans are big enough that we would maybe want to be cautious about the immune system effects? Or do you think that the restrictions in the mice are just so much bigger that we probably aren't seeing immune system deficits from something like a 1 or 2 day fast in a human?
I don't have data to support that, but I will speculate that these self-imposed interventions are not likely to have a substantial negative effect
Mm-hmm,
on the
mm-hmm,
immune system. Might be beneficial. Certainly, the metabolic benefit is real and it's important.
mm-hmm,
But I also feel, based on how we drove lifespan extension in the mice, that the interventions are not going to be a magic transformation for your lifespan. Um, and I'm going to go so far as say that I don't believe there's a little blue pill. I don't think there's one solution, because I think aging is multifaceted. It's not one thing. I don't think I don't think it's encoded in our genomes, and there's no internal clock that tells you how old you are. It's you may have some resilience in your immune function and some susceptibilities in your kidney function. So everybody's got a different set of variables that they have to address. Immune function really important. No, I don't think that the kinds of interventions that people are capable of self-imposing are likely to really impact their immune system negatively.
mm-hmm, mm-hmm. And I want to go back to sort of the frailty piece here. So some of these animals, especially on things like the 40% caloric restriction diet, they were much smaller than they would have been. They had a much lower level of not just fat mass, but lean mass as well. They were less muscular, basically. Do you have any concerns there when you think about things like things like, the new GLP-1 drugs, they seem to be having great success at helping people lose weight, but people are losing both fat mass and lean mass. Is there anything that your study says in terms of how people should think about that lean mass loss and how important it might be to preserve that?
I'm going to dodge the GLP-1 question.
Okay.
Come back in 5 years and let's ask that one again.
Yeah, I mean, that's
obviously really, really, really important question. There's too much unknown for me to. I'll speculate about some things, but not that one. But do let me come back to frailty, because frailty is really important. And I want to just describe a picture. I'd be happy to send it to you in email later. But I have a picture of two mice, and these two mice are both 4 years old. And what's interesting about a 4-year-old mice is that they're ridiculously old. Mice don't normally get to be 4 years old. We had these mice on 20% CR. And in the picture, it's so beautiful. They just happen to be nose to nose, and they're sniffing each other. And you look at the one on the right, and the coat is smooth, the fur color is black, it's a little bit of gray hair, the tail is straight. You look at the other mouse, hunched back, you can see the hind end is wasting away, the tail has kinks in it, the fur is all tatty. But these two mice are the same chronological age.
Yeah.
I look at them and I can see immediately with my eyes that the one on the left is older, biologically older. And one of the real questions of our study here is, well, how do we measure that? How do we quantify what our eyes can see? You look at an individual and you can tell that they're, I'm going to do air quotes, older in a biological sense. So two animals, the same chronological age, look to be very different in terms of their biological age. We have a simple tool. It's a checklist in our case, it has 32 items. And there's an equivalent checklist that's used by gerontologists to examine people when they have a physical exam. But, our checklist, we just go down. Do we see gray hair? Now, gray hair is not going to kill you, right? Look, but it's a sign of aging. We look to see if they have cataracts, and by the way, it's cataracts. They're not going to kill me. They're unpleasant. And we ask a whole bunch of questions, just 32 simple things that you can look at a mouse, flip them over. Do they get back up on their feet? This is a fantastic measure of biological age. I love it. And it's called the Frailty Index, and it comports with your intuitive notion of what frailty would be when you see it.
Yeah.
So, thanks for asking. It's one of my favorite tools, and we use it all the time.
And, again, as we said in the beginning, we don't want to overgeneralize results. We want to be super cautious when we're thinking about results from a mouse study and then talking about human beings. Is there any, do you think the results is there anything here that speaks to how people should think about things like intermittent fasting in their own life? Do you have any opinions on whether or not that's likely to be a good idea for most people, especially people that are already metabolically unhealthy? And do your results sort of speak to that at all?
I would say that if you are restricting your calorie input or intermittent fasting, and your goal is to extend your lifespan, you're not really going to achieve that necessarily. If you're doing these things to improve your metabolic health, I would definitely say that's a good thing. So I don't think there's no little blue pill, and there's no diet that you would tolerate that would extend your lifespan the way we saw these mouse lifespans extended. So caloric restriction, to be effective at extending lifespan, has to be extreme, and it necessarily has good and bad effects on health. And some of those bad effects are really undesirable. Being cold, being hungry, not good.
Yeah, yeah. And I mean, there's a lot of I mean, the longevity movement is really, really picking up lately. There's a lot of money going into the startup space. There's a lot of people doing very high profile things. You know, people taking what they call the optimal supplement stack and, you know, daily intermittent fasting and this, that, and the other. There's even, you know, famous people online who've posted how much their body temperature has dropped, and taking that as evidence that they're going to get a longevity boost. Do you think we know enough about the biology of longevity, generally speaking, for people to have that level of confidence that making those lifestyle changes is definitely going to extend their lifespan?
The answer is easy. You're asking my opinion, and my opinion is no.
Mm-hmm.
And I think I should be careful, but I won't at the moment, and say that I think there's a general problem in biomedical research as a whole, where there's a push for translational relevance.
Mm-hmm.
I'm going to qualify that and say research that's translationally relevant, stuff that you can apply immediately to people and improve their lives, is incredibly important, and we should place a high priority on it. But there are things about health that we don't understand very well. And I think that this is our study with mice. Most of the studies that I'm involved in are really trying to ask, well, what is the basic biology? What's really happening, you know, at the cellular, physiological, molecular level to drive these phenomenon? And as this study shows, there are sometimes some real surprises.
Mm-hmm.
And I think those surprises teach us as we go forward that, you know, if we rush to the solution, we might actually do more harm than good.
Hmm.
Again, be very careful, because there's some fantastic discoveries that have translated to, you know, cancer research is amazing, the things that have happened. But I think that in aging research, I'm going to say these things again: don't smoke, exercise, eat right, sleep, enjoy life, love your family.
Mm-hmm. Mm-hmm.
Those are
Yeah, and I
things
think,
we really do.
yeah, I mean, I think, you know, to riff on what you were saying, you know, we know more than ever. We've got more studies we can look to than ever when it comes to this stuff. But it's important to remember we don't know everything. In fact, there's still so much more we don't know than we do know, that we should, I like to remind people, and myself really, you know, we need to stay very, very humble, because even though we know more facts about the world and about biology than ever, you know, the space of knowledge we have is so much smaller than the space of ignorance, that, you know, we should be cautious about thinking we know how to engineer the optimal life or whatever.
Yeah, the iceberg metaphor is overused perhaps, but this is it. I mean, we know what the top 20% of the iceberg looks like.
Mm-hmm.
We shouldn't fool ourselves to think that we see the whole thing.
Mm-hmm.
And I think for me, this is job security. It gives me, I like learning. I like uncovering things. And there's, you know, essentially endless discoveries to make. And
Mm-hmm.
they're hard, and they take sweat and labor, and 7 technicians in 8 years
Yeah. Is
put
so
them.
yeah, I would love to ask you about that a little bit. So, like, you know, you had to have a staff, you know, a fairly sizable staff, and you had to have 8 years of time to do a study like this. Obviously, you know, it was worth it in the sense of, I think anyone interested in this stuff and in this field would say, you know, this is a great study. We're learning a lot from that. We need to be doing this type of research. But, you know, is there something special about the Jackson Laboratory and where you're doing this research that enabled you to do a study like this? Because, you know, a brand new assistant professor somewhere at a big shot university can't wait 8 years to get a paper out. So what enabled you to actually do a project like this?
I think that the short-term funding cycles are a special challenge for aging research, especially if we use a model organism like a mouse that can live 3 or 4 years, and we have to stagger the starts. The Jackson Lab has a whole community of people who have all kinds of expertise in all aspects of biology, but there's a particularly dense concentration of people who know about mice, know how to handle mice, and know how to phenotype, how to characterize mice, and, you know, and determine their health. So I was really lucky, and the staff members were just wonderful. One of my team members did over 5,000 frailty exams. We had 100,000. No, wait, we must have had hundreds of thousands of body weights, and people were dedicated and they're
Hmm.
careful. And it's really important to be careful in research. You want to put the numbers in the right column, you
Hmm.
know. And the statistician that I had working on this project and the new statistician who came along and replaced the first one are just wonderful because they look at the quality, they fix the mistakes. So there's a lot of effort that it takes to do research. And you have to pay people salaries, you have to pay for the facilities and the experiments. It's expensive.
Hmm.
I feel really privileged. I feel really privileged to be able to do it.
Hmm.
Society has given me and many of us scientists has given us this opportunity. And I want to express my gratitude. And hopefully I'll return something.
Yeah. Yeah. Well, I mean, yeah, you've returned this, and then, I mean, this is a wonderful paper to read. It was, just a massive undertaking, and people seem to be responding to it quite well. I think a lot of people were excited to dig into this one, just given the number of mice, the amount of measurements, and just all of the stuff you packed into this. What's coming next for you guys? Are you following up on this particular study? What are you working on now?
I remember I said I was a statistician,
Hmm.
and when I was an undergraduate, I talked to my statistics professor and I asked, "Why are you a statistician?" And he said, "Oh, it's because I can dabble in a little bit of everything, because everybody has data, and I can work on this and I can work on that." And I feel like I would like to move on from this. I think there are some really important results that should and can be followed up, but they require a different skill set from what I have.
Hmm.
Um, and I'm going to say it. Where we're really interested in looking next is related to stress resilience. So there
Hmm.
are all kinds of stressors that people experience in life. And one that I'm particularly interested in, and it has the potential to affect half of the human population, is reproduction, pregnancy, birth, lactation. Um, I haven't talked to a woman who said it wasn't stressful. And when we study this in mice, we can see how pregnancy and birth have short-term and long-term health effects. And I feel like I want to understand better what are the long-term health effects of just in mice, multiple pregnancies, and where does this go? Does it?
Hmm.
Yeah,
Hmm.
I think no matter what the answer is, it's going to be interesting, and you'll be talking to me in 8 years because that's how long it'll take.
um,
I'll miss
any we've
you.
Yeah,
Time.
we've covered a lot. I want to thank you for your time, Gary. Really interesting paper. Really loved reading this one. Any final thoughts you want to leave people with about this stuff, or anything you want to reiterate about what we talked about today?
No, I I have a couple little notes of the things that I really wanted to say. You want to live a healthy, happy life, and there are well-known prescriptions for doing that. And don't go to extremes. And understand that a study like this is intended to unravel fundamental biology, and it's not intended to give you a recipe for living longer. Um, so I think that understanding that biology has the potential ultimately to give us better prescriptions for living longer. As we go on, we learn, we do better. But please don't go on extreme caloric restriction.
All right,
Thanks.
Gary Churchill, thank you for your time.
Thank you so much. It was a pleasure.